Okay, folks, let's start. So I will just point out one of the things that I could not discuss about your exam one, because there was a makeup that I had to give for that. And we're not going to review the entire part, and you know that, but I'm just going to point out a couple of things in here that would tremendously help with your next one. So if I just look at the quiz, like this is your exam one stat, essentially. So let's take a look at this, and this will help with you a couple of study patterns. Give me one sec right here. Yes. All right, so here you can see that you have a 98% of a score, and then I have one of the question that I'm just trying to find out. Where is that? Let's see. All right, so here, this is one of the interesting thing is in here. If you look at this question, that the phenomenon which a chemical absorbs light at one wavelength and emits as a specific and shorter wavelength, and look at the percentage that missed that question. So 48% only is correct with that. So anybody, anything rings a bell in your head? That I said that's like almost 50% people miss that. So I brought up the zoom in here and see this. So just that one question tells you that there is a lot of information in there, and if you just use a short period of time, so you can do this with your all exams. So what I did is, like, I not only opened up your quiz, I opened up all your practice quizzes as well. So you look at the question and then just do a control F or just type a word. You will see 70% of the question you have seen, some of them line by line. Like I did the questions, those questions are just copied, not even the values were changed. Even if I just go onto the if you just remember, you can just go by yourself, that the Km value, that 2 millimolar multiplied by 36 millimolar, that is on the power on the zoom. So I just used those values which were in there. So essentially, you just keep practicing those or study every day a little bit. If we go into a long-term memory, you will not forget when you come to the house. So that's essentially, you do not have to study 4 hours, 6 hours every day. I do not expect anybody to do that. But the more you see, rather than just only the weekends or a few days before the exam, that will actually help. So what I would suggest that you have your group me or everything, and for your practice quizzes, I do not put all the questions in there because there is 15% point in there. Like people, they call it as a great flop, but that's okay. 50% there in your practice quiz, 50% on your quizzes. So exchange, take an Excel file or a Word file, whatever works, and then exchange the questions that what you have seen, your friends have seen, and take those questions back. And as I said, that some of the questions which are missed by the most, where I have changed, some of the questions are exact the same way, the answer choices. And the questions, as soon as I change some of these answer choices, that actually is missed by more number of people. So that's why I say that if you just look at the blurb before you start your practice quizzes, says that you look at every answer and try to make an incorrect one a correct one. So you just write it down. Every question you cannot do that. I completely understand that. There are some of the questions you cannot do that. If you just have 4 choices, you have to bring in something. But the questions that you can rewrite, so think about that. And if you spend that much extra time, that you will see that drastically your grades will actually improve. So that's essentially what I just meant to say by that. Say for example, and then say for example, there are some of the questions. See that I said that. See that this hybridoma, and I will just explain that is what is happening in here. So that 20%, which means that only 20% actually got it right. So the thing is that this is a difficult question, but we have seen a similar type of question. I will explain that what it means. that here we see that there is a DHFR, which actually is for the de novo pathway that is also mutated. So, and you have the B cells that is fused. So the concept that if you're missing A minus or AGPRT minus, that is supplemented by the B cell. Same, any other gene like a DHFR was missing, it will be supplied by the by your B cell. So essentially, what will happen that your myeloma will not grow without nucleotide through the Savage pathway or the de novo pathway, because de novo pathway doesn't have a DHFR. You have supplemented nucleotide to grow. Once it fuses, you have all DHFR TK minus, AGPRT minus negative myeloma cell. Then for your B cell, all are positive and it is mortal. And for the hybridoma, all are positive and now it gets the mortality. So now the media, see that what I did, the only change in the class when we used an AT media and there was no DHFR mutation in there. There was AT media, so aminopterin was blocking that pathway. So now what happened that I do not have aminopterin in there, so the myeloma cells doesn't have the DHFR, so it will not be able to use that pathway either. On the other hand, as I have H and T both, the Savage pathway should work if they have AGPRT and the TK. And the B cells and the myeloma cell, B cells and the hybridoma cells both have that. B cells will eventually die because it's mortal, and the hybridoma cells will be able to use this one, and it will do. So this is basically what you do is like you draw the myeloma cells, you draw the hybridoma cells and the B cells, and then see that what is complemented. So for myeloma cells, DHFR minus, D minus, TK minus, AGPRT minus. For B cell, it is all plus. For hybridoma cells, it's all plus. Only difference, this one is mortal, this is immortal. And see now that whether they have the ingredients to use the first pathway or the second pathway. So this is how your thought processing. And the reason that again, not every question is like that, but why you have a few of these questions that actually will help you in some of the competitive exam, professional school, where you want to go in there, that how you need to think about these questions. And again, everybody is not going in there. That's why you see only one of those type of questions. I'm not going to put every question in there. That pretty much, if you look at that, 70% of the questions that you have seen and very straightforward. And if you just spend a little bit of time, nobody should fail. Everybody should get at least 70 or above. If you just put a little bit of extra time, you can see drastically your grades will increase because you have 30% grades coming from which I can get 30 out of 30, 100%, almost 95 or 100% you can get in your practice quizzes and quizzes. So which boosts for exam almost 6% of your grade automatically. So these are some of the things that I will say. I will just not go into much more detail compared to other than this, because we do not have that much of time. But again, if you want any explanation of any particular question that we have, just all you have to do, you can just do it over Zoom. You can just during their office hours, or if you're just coming for office hours for the second exam, I can happily explain any of the questions. So it's all open now, and you can do that. So let's go to the material. Any questions that you have? So, so one of the things is that the most important thing that I would say out of all is that as the material is vast, it's a lot of material. Even the star power slide and the highlighted slide, those are so. Make sure that you look at the material often and look every day. Even if you can spend 20-30 minutes every day, that will actually help a lot. All right, so let's start with the cytoskeleton. We stopped at the profilin tyrosine polymerization. We stopped at this one, and in the motility, how this is happening. Now here, we will start with this. So here, this is a cell. I mean, the white blood cells are the motile cells that moves from one place to the other. There are other cells in the, like some of the osteoblast and osteoblast can also move. But there are some motile cells in place, and what we were seeing that there is lots of these actins which are cross-linked against each other. So how this cross-linking or the branching happens, because so far we only talked about that they are in a filamentous form, but how a branch can form. And that is what is pointed out in this slide, important slide. A couple of informational slide. A lot of detail is in there, but we are not going to go into that. So just remember the name of that. This is called an ARP or ARP-2 complex. Remember that sometimes people mix this one with the ARF, which along with the SAR1, which is a GTPase that goes into the membrane as a scaffold to make the vesicles. So this is entirely different. So this is ARP-2-3 complex. So you have 7 subunits with that, and just only 2 of them are shown, which are medically bound. So these molecules are in there, and these are essentially some of the side branching nucleation molecules. And for that, first, what you have to do, that there is a different protein, look at that, a different protein, which is the NPF, the nucleation promoting factor. This WCA, these are different protein for that detail you do not have to. But a nucleation factor right here must come together, and it must recruit 2 molecules of globular actin. Because I already saw before that if there is a nucleation of these the actin molecule, the polymerization goes very fast. Otherwise, it takes a little bit of time. So that is a rate limiting step. So here, there is just not only just regular globular actin, there are some protein scaffold, and this protein scaffold is nucleation promoting factor, NPF, that recruits 2 molecules of these globular actin. Once this is actually done, then you can see that in between these 2, the ARP-2-3 complex, which is right here, is being shown, that is recruited. So you can see that one globular actin connected to one end of the nucleation factor, and the other end there is one ARP1. And then there's another nucleation factor at the top. You have the globular actin and the ARP-2-3, another ARP-2-3 complex is connected there. So these are the 2 units that are essentially in the middle. So remember that these 2 individual dots are not 2 different proteins. This is just one complex, just right here. 2 subunits, dimeric subunits are 2 and 3, and that is in the middle. And that is essentially the scaffold. You can see right here that this can start a branching. It can bind to the side of existing filamentous actin or F-actin, and on top of that scaffold, you can put a side chain. So other than that, the globular actin cannot bind on the side of an actin. So just remember that branching of. So what essentially you are seeing that sometimes we can give you a example of that. If you do not have an NPF, suppose there is a mutation in a person, and they have an NPF problem. What could be some of the outcomes? Say, for example, some of these people have less motility of the white blood cells, which can increase infection. So remember that, connecting a couple of things. White blood cells need movement. Actin is responsible for that, and which can increase the infection process. They could be immunocompromised to some extent. And branching of octane, which is essential for the leading edge of a motile cell, motility of the cell will cease, and we can just make a story out of that as well. So just to remember that how we can put those in context. All right, so these are some of the electron micrograph of the branchings. And now we are looking at that how these cells actually change directions as they move. So essentially, many of these cells are attracted to some of the chemotactic factor. These are released by cells or the pathogens. There are many different things, and chemically, the cells can sense those chemical gradients and will move towards them. And if the source changes, it can change direction as well. That is a part of a signal transduction. We are going to say that in the later part. So the signal comes into the cell, and this is the mechanism of movement. So see that what is happening here. So this is a minus end. Remember that barbed end is a plus and the pointed end is a minus. So now the cell, the cell was moving on this direction, right here towards the arrow. And now, say for example, instead of moving into this direction, it now wants to move in another direction, and what will happen? So let's first see that what is essentially happening when it goes into this direction. First, you can see that there are some filamentous actin, and it is also cross-linked. This is an ARP23 right here, ARP23. We are going to see that the filament is some of the cross-linking stabilization molecule. It actually helps in stabilization. There are a couple of slides before that we have seen the chart. If you can see right here, filament is helping in cross-linking as well. And then for the stability, there is also another protein which is called FC and the tropomodulin. And nebulin, tropomodulin and nebulin, we are going to see it a little bit later. But see that we will see the FC. So right here, you have these yellow ones, which are the capping protein, and these are your FCs. So when these ends are actually capped, that filamentous actin will not grow any farther. It is stable and it will not grow any farther. So this is basically end of it. It cannot add it. On the other hand, this plus end is open, so you can actually add the ATPG actin right here. So what has been shown, let's say for example, this is right here, treadmilling happening. That means that ATPG actin can be released from here, and see the profilin actually charges it with the ATP, and then ATP bound actin can be added in there. So on the other hand, if you see that this is the part right here, the gray part, the scophiolin and the gelsolin, these are the savoring protein. If I just go back a couple of slides, if you just look at that right here, the length, profilin and the gelsolin, and then the next part we had already saw the profilin, and gelsolin is another one which actually essentially shorten the chain from the minus end. So a lot of different protein, and each is different things. So just remember that what exactly each one doing. Capping protein like a CAPC, filament cross-linking protein. The savoring protein is profilin gelsolin, and profilin is adding up the charging the ADP actin to ATP actin, and it can be added to the plus end. And ARP23 that help in the branch. Now what happens that if this cell is moving on this direction, and if it wants to change the direction on this, say the arrow is on this and it is not moving anymore, what it will do, that it will cap pretty much all of these, essentially, cap all of these with the capping protein, so that the actins will not grow in this direction, and eventually they are going to get depolymerized, and new actins will grow in this direction. So this is where the cell was going, and if the cell wants to go in another direction, all the cells, all the actin molecules are going to be polymerized in this direction and grow, and these will essentially cap and it will shrink. So this is how they can essentially move. So let's do a quick recap of some of the things. It's very straightforward. 1 minute. Let's go forward. Good, everyone? Last chance. It's good enough. All right, stopping interface. Yeah. expected a little bit more. All you have to do is like to look at the previous slide. Anyway, so this is coffilin and gelsolin, which is the previous slide right here. If you just look at that, the coffilin and the gelsolin, these are the ones which are from the minus end that are depolymerization happening with that. So again, I just went up there. So the length, coffilin and the gelsolin, they decrease the length, and this is where you were seeing them in action. All right, so this is the question that, again, this is another one. I will give you a little bit more time with this. This is the one that I just talked over. So if you just understood that. You guys are good, I guess. That was pretty much all of it, I guess. Anyone else? Probably not. So I'm stopping it. Yeah. Right. Yep. That is correct. So that's all you have to just understand in this process is that the capping protein like Cap Z, that was basically the previous leading edge that as I said. If we just go back in here, see that this is where it was going. So you do not want the cell to go in this direction. So essentially, all of these will be the capping protein on Cap Z will essentially block these. On the other hand, the other choices where that this is the new direction. If the cell is going in there, you do not cap those because they need to grow. If you just cap those, they will not go. So capping protein is a Cap Z plus end of the effect is new leading edge. No, because it is going in this direction. You want active molecules to be added on the plus end. And the other one is the tropomodulin minus end of the effecting of the previous leading edge. You want them to be coming off. So capping anything at the minus end does not work because you want the minus end, the coffilin gelsolin, they are removing those so that these are available and can be added to the new direction. And the other one was cap minus end of the new leading edge. Also, you do not do that because you want the treadmilling process essentially. So see that right here in this direction. If it is going in this direction, you want things to be coming off and added to the new directions so that they can actually grow. So never cap the minus end. You are only going to cap the plus end of the direction where the cell doesn't want to go. And that is the only answer that you want to have. All right, so that's good. All right, so these are some of the very self-explanatory things like the Listeria monocytogenes, why it is so difficult for your immune system to actually take them off. Because they, and antibodies and your immune cells do not see them if they are only inside the cell. So what they can do, that they can go inside a cell and then they actually polymerize the actin tail. It's amazing. So basically what is happening, you can think about that you are basically building a stair and climbing up the stair at the same time. And it just happens so fast that these actin tails actually propel them, and they can actually make these type of protrusion from the membrane. And this membrane breaks it up, and this whole structure can be transferred to another cell. So the listeria never comes out of the cell when it's infecting a neighboring cell. And that's why sometimes these infections are super dangerous. So here, the intracellular listeria that essentially you are seeing this comet-like green-like tail, that is the active polymerization behind the bacteria. The bacteria is at the very top in here. So there are some amazing videos. There are some couple of videos that you can see. So take a look at those as well. So it's essentially what is happening right here and how it does that. So listeria contains a protein which is known as ACT-A on its membrane, essentially. And these pink ones are the ACT-A. So the ACT-A essentially is an active polymerization protein. So it can add proteins to the branching as well as polymerizing from the branches. And this is essentially what is happening. And this is an extremely potent polymerizing agent for actin. So what you are looking at, that this is the bacteria. At the bottom of it, you have these actins. And all these active protein, they essentially attract all the cellular globular actins and put them under them. And essentially, these are depolymerized from the minus end, and they are just going fast. And this is extremely fast. It can actually go on in this process. You can take a look at a video later at some point of time. I think that it is in your supplementary videos. You can take a look at that as well. This is also another last part, that how much extent these active polymerization can actually alter the cell shape. So here, what you are looking at, that you are looking at a platelets, thrombocyte, and this thrombocyte is essentially coming near a clot. And this one is a protein called GP1B9, which have this cross-linked filament, and these are some of the actin filaments. So when there is a signal that there is a blood clot, what happens that near that clot, these platelets actually home in, and they will try to extend themselves. So this is the initial structure. This is an activated thrombocyte, and see now the thrombocyte extended itself like this, and this is like from a pizza dough to making a pizza, essentially, by the help of an actin. And it will basically seal that, and this will essentially seal that clot so that there is no fluid is leaked out of that. So this is a GP1B9, which essentially binds itself to the clot, so that this is essentially an anchoring protein. So this protein anchors the thrombocyte or the platelet in here, and now this is a signaling molecule as well. This signals the cell to extend its actin filament, to stretch the cells. So basically, you have the clot, and at the bottom, you have the cell that basically acts as a piece of band-aid, that you can think about that. That's essentially what is happening. And here, this is just only for your understanding in here. We are going to learn this in a different unit, the next unit, that these are the signals from soluble factors, other cell extracellular matrix. And then these are the receptors. Here, actually, GP1B9 is a receptor for that. And then you have the signal transduction pathways, which model this cytoskeleton, organizing and movement of organelles, cell shape movement and contraction. And this is essentially how it happens. It is not easy to give the cell a signal. Otherwise, just by itself, these modifications doesn't happen. Even for the cell division, you need a step-by-step signaling process that we are going to see. So that's pretty much the part one. So we will start with the actin binding proteins, and then we're going to look at some of the intermediate filaments. So this is the same picture right here. So we already talked about the polymerization. That means that you are adding to the plus end profilin, positive regulator, tyrosine beta 4, negative regulator, length, basically severing the length, cofilin, gelsolin, ARP 2, 3, 4, nucleation branching, or and then basically if you just start a branch anywhere, we already saw that. And cross-linking is filament. So what is the difference between that? This is a subtle difference, remember that. ARP2-3 complex is allowing the branching, and to keep those branches in place so that they are structurally rigid, the filament actually cross-link them. And now we are looking into the motor proteins. We already saw CAPZ. We are going to see the tropomodulin. We will see something like a titin, nebulin, troponin, tropomyosin. These are the ones that we are going to see. So the first one is motor protein. So these motor proteins are these are amazing proteins, and they actually move things from one place to the other in the cell. And if they are fixed, they help in contraction, particularly in muscle cells, essentially. So the myosin 2 is the most common one. There are more than 20 different types of myosin, and all of these myosins are associated with actin. When we go to microtubules, we're going to see that there are some kinases and dyneins, different proteins associated with microtubules. So this myosin 2 is the most common one, and the generalized structure is that they have a head-like structure, golf club shape, and then they have a neck, and then they have a coil, rod, or tail domain, essentially. And this region, when the neck, is also known as the hinge region. And sometimes there could be, it's a multi-protein complex, so that you can see that this is dimeric. You have one in olive color, one in green. So there are two molecules of proteins coiled again in the tail region. Those are the two heavy chains. You can see that those are the two big chains. On the other hand, these rings that you are seeing in black, those are some of the light chains. And all of them together make this myosin molecule, essentially. So electron micrograph, these could be so big that in electron micrograph you can pick up this essentially. So most abundant myosin is essentially the muscle myosin that you can see everywhere. So a couple of different ones. So here you have the myosin 1. You have a head, neck, and the tail. There are some calmodulin light chains. This calmodulin is a calcium binding protein, and calcium is one of the most important regulator of these cells. Then you have a myosin 2 that you saw in the previous picture. Then you have a myosin 5 where you have a coil, and then you have a separate tail. And see that right here, calcium binding protein, calmodulin light chains. So these are regulatory protein. These are always in this neck region. So these are myosin 1, 2, and 5. Just remember this, that you will always going to get this question in your quiz or as well as in your exam, that there is one myosin which actually moves towards the minus end of the actin. Remember that your actin has a plus end and a minus end. All myosin motors, when they are moving, they move towards the plus end of the actin. The only exception is the myosin 6, which moves towards the minus end of the myosin. So just remember that. So 1, 2, 5, everything, the others, these are the most common ones, all move in the plus direction. So now what we are looking at, that this is essentially the ATP hydrolysis of the myosin head that happens in the muscle, essentially. So what we are going to see that the muscle essentially does have, I will just take a look at one of the picture from here, and I'll go back. So when you look at the general structure of the muscle, we are going from here, right here. This is a particular part of the muscle, and it's divided into skeletal muscle. These are divided into units, and one contractile unit is called a sarcomere. That's what you are seeing right here. And if we just take one of these, essentially, that's right here, this is essentially a myofibril. And then if we just take one of those, that you see a myofiber, which is essentially a muscle cell. So this is an individual muscle cell, and of course you can see that they are multinucleated. They develop as myoblasts, single cells. then they fuse as they develop, and they become myocytes or the muscle cells. Myo is, there are two words that we are going to use, either myo or sarco, S-A-R-C-O. These are the two adjectives for muscle related. So say sarcoplasmic reticulum, that's the endoplasmic reticulum of a muscle cell. Sarcoplasm, that is the cytoplasm of a muscle cell. And same, myofibril is a muscle fiber, myofiber, muscle cells, these are also same. So now in each of these cells, what you have, in the middle, you have these myocytes. And these are the myosin head, and this is the rod portion. So you can see that these myosin heads are directed on this side, and the rod portion extend into the middle. Here the heads are on this side, and the rod portion is in the middle. So these are known as the thick filaments in the muscle. On the thick filament, you are also going to see there are some thin filaments in association with them. So how it looks like this, that in a muscle, if you just take in the middle, you have this thick filament. This is your myosin molecule. And on each side of the myosin, of course, it's a three-dimensional structure. These orange parts are the actins, essentially. And see that this is the minus end of the actin, and these are the plus end of the actins. So it's what happens that we already know that all these myosin heads, most of them, particularly here it's myosin 2, they move towards the plus end. So what happens that when a muscle actually contracts, each of these heads will bind to the actin and will try to essentially crawl on top of the actin. So in that process, it is trying to crawl on this. These are trying to crawl on this, and that in that process, this actin is moved towards the center. This actin is moved towards the center. And as the actin is bound to some of this plate-like structure on each end, which is known as a Z-disc, this entire purple plate is going to move in, and this gap is going to shorten. That's essentially the muscle contraction. And then when ATP comes in, it can release these myosin heads, and then the muscle will relax. It will go back to, but due to its elasticity, there are some elastic proteins in there. It will go back to its extended state. So what we are seeing in this few slides, on the next slide that we're going to see, that how these myosin heads interact with the actin and actually pulls it towards the center. That's essentially the part of the story. So let's take a look at this essentially. So here you are looking at the thick filament myosin head, and here you have the actin. So this state is known as a rigor state, and there is a site where ATP can bind. It is empty. In this condition, the myosin head is bound to this green globular actin on the filamentous actin. Remember that it's two chains, they are close together. Now, the ATP comes in and binds to that. And ATP immediately dissociate the actin, the myosin head from the actin. Now what happens that this ATP is hydrolyzed into ADP and PI, and at this point, the ATP is still bound. When ATP is still bound with the PI, this head actually pivots and binds to a new actin molecule. So from here, it now binds from there to this plus end. Remember that this is plus end. So this was from towards the plus end. That's why we say that it goes towards the plus end. Now, in the subtle reaction, the phosphate is released. Still, it is ADP bound, but the phosphate is released. The head pivots and moves the filament, and this is essentially the power stroke. It basically, so remember, in between the 2 and 3, remember this very, very carefully. When the ATP is hydrolyzed into the ADP and the PI, right here into the ADP and the PI, it just binds it, but it does not pivot it forward. When the phosphate is released, at that time the flexion happens. That is the power stroke. So we just give you the thing is that when ATP and PI binds to that, there is a power stroke. But just remember that the phosphate release and the ATP is still bound, the head pivots and moves the filament. This is the power. This is binding. ATP and PI just binds to a new subunit, but the phosphate release actually gives the power stroke. So this is very important. So know this thing. And this is known as a cross-bridge slide cycle. So it's in your blood, but you can write it down that what it is. And then the ATP is released and it goes to the rigor mode, which is this. Now you can tell that where the after death, after about 3-4 hours, usually the body stiffens up. And what is that called? Yeah, so that's where the rigor mode is actually that comes from here. Essentially, what happens that all the muscle ATP and hydrolyzed deplete, and essentially you go into this state for the biggest, and the muscles become stiff. This also happens that you have seen these like high intensity games, but basketball you've seen that sometimes, it even happens in football. But some of the players suddenly had a cramp, and they need to be carried out with the help of something. They just cannot even put their feet there, and that is essentially a physiological rigor condition. What happens that due to the high intensity of the muscle usage, they deplete that muscle. At that point, the muscle cannot release the myosin head from the actin, and it is in this rigor state. The only way you cannot just do, you just have to hydrate and let the muscle generate the ATP so that it can release that. There is no medicine that you can just inject that will immediately. That is the only way that it will just take care of itself. So you know that what it is in this process. So remember that it is the calcium is required. All of the myosins move in the plus direction. The exception is the myosin 6. And this is essentially known as the cross-bridging cycle in muscle contraction, essentially. So let's take a look at how essentially myosin 2 actually moves. And when the myosin actually moves, if you increase the neck region, it increases the step size of it. So what we are seeing that this is your velocity per micrometer per second, and this is a normal wild type myosin. I have added 1 extra amino acid in there, and it increases the step size. 2 extra amino acid, increase more. 3 more. So if you have the more amino acids added to the neck region, it can essentially increase that neck size. So we're going to see some problems out of these as well. So let's take a look at this, that how the myosin actually moves, and the movement is essentially what we know now very, very well, is that there are 2 hypotheses that initially proposed, that the movement could be hand over hand, that you are just moving one hand, then a hand. This is one of the movement. Or another could be that, which is a permanent leading edge, that you move, then you scoot the handbar, then you move and then scoot that the back leg behind that. So to do that, what the scientists actually did, that they label this myosin with a fluorophore, and this is a single molecule fluorescence microscopy. And then they looked at the data that during the movement, how much they are actually moving, and what they saw that essentially the dimension, that each step of these particular myosin 5 is a 72 nanometer step, pretty much. And from that, they actually concluded that this must be an alternating leading heads, which is hand over hand, essentially, because if it is just the permanent leading head, just scooting the leg behind, it would give it touch. So I have no idea. So essentially, yeah, this is. That's fine. So essentially, what is happening is if it's a permanent leading head, the length of this is half, which is not shown. So if this was true, it would have been predicted as 36 nanometer, which is correct, 72 nanometers. So we know now that it moves alternately leading heads. There are a good number of some of the extra resources in there, and you can just take a look at some of the supplementary videos. Some of them are long. You can just take a look that there's some interesting material as well. So here you have the myosin 5, how it steps over the period of time. So each of these dots actually denote that what they are essentially doing. So essentially what they're doing, that at each step, why you are taking multiple measurements, because this is like a Brownian motion. They're just trying to go and then step forward. It is not that they just go like us. So it is just oscillate in that direction and they can go farther. And each of these increase, remember that when you are going towards the x-axis, that is not the step. When you are going, the y-axis is the step, and all the steps are not the same. See that some of them can be a little bit big, some of them could be a little bit smaller. And again, you are looking at a single molecule and taking the data and experimental data. Every data will not be the same value, so just understand that. But if you see the average value is 72 nanometer and nowhere close to the 36 nanometer, and that's how it has been. It has been decided that this is basically, that was a conclusion essentially of the experiment. That's hand over hand process. So here we are looking at these different class 1. You can see that you have the step size 10 to 14 nanometer, myosin 2 about 8 nanometer. This is the construction of the muscle, whereas at myosin 1, the myosin 1, they do the membrane-associated endocytosis essentially. So invagination of the membrane is also caused by this. And these are the myosin 5, which can move different vesicles. So now you know that essentially, remember that we talked about, so these are some of the puzzles that will come together. We talked about that how the pinching of the vesicles happening, where is the address, where it should dock, but how from one place to the other. If one is carrying it, it's just not diffusing the cytoplasm because it's too dense. It cannot. They are carried either by the myosin motors or dynein or kinesin motors. There are small alleyways. These are made up of actins, and then there are superhighways. Those are made up of microtubules, and it's an amazing way that just take a look at some of the videos. It will show that the kinesin and the dyneins move things much faster. They are like fast cars on the freeway, where the actins can precisely deliver where they need to. And step size is 36 nanometer, and if they go one at a time, 36 by 2, 72 nanometer is the step size, because it goes in this directions, alternating leading heads. All right, so let's take a look at a problem that how we can do this. Logic. What happens at amino acids? These are the thing that the graph that you're seeing, that is the graph from the recombinant myosin with the 4 amino acids. That's not the wild type. That's the myosin. That's good enough. Anyone else? Okay, stopping it. All right, so let's, I'll build an answer. So let's take a look at that. So first of all, when we're looking at these myosin 6, if you just go to a couple of slides back, myosin 6 is the only myosin directed motor. Rest all are this. So myosin 6 moves towards the myosin. So the first thing that what you have is just pure memorization is myosin 6 towards the plus end of the actin. So AB immediately goes out of question. So it doesn't move towards the plus end. So minus. So the question is that this is what it is. You have added 4 amino acid, and at that time you have the 73.75 nanometer. It is step size. So here the idea is that if you just go back to a couple of things right here, when you add amino acids, is step size increase. Everybody good with that? So the recombinant, you have added 4 of them, so step size increase. So what it means that the wild type, the value of the step size must be less than what you have. So 73.75 is here. It cannot be more than that. It must be less than that. And let's take a look at this once again. It's 70 nanometer steps towards a minus n. This is 73.75. 70 is less than that. The other one is 75. So you have added 4 amino acids to the recombinant one. The wild type should be shorter. The wild type step cannot be larger than that. So that's basically the logic. So the 70 nanometer, that would be the correct answer essentially. Yeah, makes sense? So this is how you can analyze those questions as such. The first part is just a pure idea that it goes toward the minus n, and then the logic that why it is smaller than this, because this is the recombinant one, which is longer. All right. So we already talked about this, that what it is, and we've talked about the general structure of the skeletal muscle, and we already talked about that. And let's take a look at a couple of names, and these are kind of like a pure understanding of these region. So the first thing that each of these zones, contractile zones, are known as a sarcomere. And each muscle, it could be extremely long. I mean, like there are some of the sartorius is probably the longest muscle in your wraps around your thigh, which is like 2-3 feet long, essentially, depending upon your height. But these sarcomeres go from one end of these myofibrils to the other end. But they can be divided just for, again, they are not divided as such, they are continuous. But just for our understanding, we can divide these in structural units or contractile units. So on each side, you have this Z disc. This is essentially round. Remember that we are looking at it in two-dimensional structure, but they are essentially round. They are like this. So this is essentially one Z disc right here, and there is another Z disc right here. These are the actins and H zone right here, and let's take a look at that. Right here, this is called the H zone, and this is right here. We call this as an I band and the AI zone. So what are the different things that you need to remember from here? Just remember that next to the Z disc, the area of the actin that is not overlapped by the myosin is known as an I band. So, right here, this only reddish part, that is your I band. On the other sarcomere, this is also the I band, because there is no blue in here. Non-overlapping myosin with actin, only actin in the I band right here. On the other hand, the A band is just the length of the thick filament of the myosin. So if we just go back one end to the other, right here, this 325 nanometer average, this end to this end, this is the length of the myosin. And myosin or the thick filament right here. That's your A band. And then in the middle part of the myosin or the thick filament, the area where you don't have any overlapping actin, that is your H zone. It's only the rod portion of the myosin. So these are the three things. Remember that A band, I band, and the H zone. Now, see that what is going to happen during the contraction. During the contraction, just draw it right here. This Z disk is going to move in this direction. It's going to move in this direction. The reason is that these things are going to bind and move, and this will overlap. So basically, the I band will essentially shrink as it moves. So I band right here will gradually, during the contraction, will disappear. And the H zone also will essentially shrink and almost disappear. So that's essentially because the overlapping of this will increase. But the A band, which is the length of the myosin or the thick filament, that will never change. So just remember that. That is very important to understand that your A bands never change over the period of time. So let's take a look at a couple of other proteins that how they are involved in that. So CAP-Z binds to the plus end, tropomodulin, nebulin, titin, troponin. So this is self-explanatory. Let's take a look at this. So here you have the 2 Z disc, and this one right here is the minus end of the actin filament, minus end, and tropomodulin is the capping protein of this. Whereas the CAP-Z caps the plus end. Remember that? Plus end, this is your plus end. So, and in the middle, you have the myosin thick filament. There are some other proteins in there. So essentially what happens that this is the most important part. Remember that we were talking about this actin remodeling, some of the myosins that move on these actins. In the muscle cells, no matter whether it's a skeletal muscle, smooth muscle, myocardium, like a heart muscle, in the myosin, the structure of the actin and the myosin do not recycle. They are capped and they are absolutely stable. So you can increase them in number as you can just work out, or you can increase your muscle mass. Your muscle can go out, they can add more cells in there. But if you have an existing muscle, yeah, if you can tear a muscle, you can recycle, that's different things. But in a normal muscle cell, these actin and the myosins never get recycled or degraded. They do not polymerize and depolymerize. They just act as a contractile agent. And why this is so? Because their plus and the minus end are capped, so nothing can come out, nothing can be added. So their lengths are fixed. On top of that, you have some other proteins. So you can see that on the side, you have these proteins called nebulin, which actually doesn't allow the filaments to break off from the middle. So again, CAP-Z as well as tropomodulin towards the end, nebulin on the side, and see that this protein is titin, which binds the thick filament or the myosin with the Z disc. So what happens that once the contraction is done, ATP comes in and allows the myosin head to release. These titins essentially helps them to go back into the extension stage or extended stage essentially. So what will happen that these will bind to these essentially, and when it is allowing, when ATP comes in, all the heads are released from the actin. These titins are essentially going to extend, and they are going to allow the myosin to go back to its original position. And this middle portion is also known as the M line, which is essentially there is a protein called desmin in the H zone that's in there. So how this cross-bridge cycle and the whole thing happens, and there is a regulation of that. So know this regulation. So we only talked about that in this process as well, right here, that the hydrolysis of the myosin head, how it moves. But this is essentially the regulation that is needed in this process. So each of these actin filaments, they are covered by this filamentous structure. And this is a cryo-EM microscopy. And this is essentially tropomyosin. Remember that there are 3 dates of tropos, so make sure that you make some cards and look at them, each one of them. You have a troponin, you have a tropomyosin, you have a tropomodulin. Tropomodulin actually do not modulate. Instead, tropomyosin does that. So what tropomyosin does, in a relaxed muscle, it actually binds to the binding site of the myosin. So again, I'm repeating, on the actin, there is a certain position in which the myosin binds. The tropomyosin covers those binding sites in a relaxed muscle. And on the tropomyosin is also attached with another regulatory protein, which is troponin. So troponin right here, the green one is the troponin. And the function of the troponin is to allow the tropomyosin to either cover or uncover this binding site. So what happens that when a neuromuscular signal is received from the neuron, there is an action potential that goes on the muscle cell, goes into the ER or the cerebrospinal reticulum. Not talking about detail, but essentially what happens that calcium is released from the endoplasmic reticulum reserve in the cell, and the calcium actually binds onto the troponin, and this is a trigger for the muscle to contract. So when the calcium binds to the troponin, then the troponin actually changes its conformation and it lifts up the tropomyosin, exposing the myosin binding site. So here you can see that troponin is covering those red dots right here. And when the calcium is present, this is minus calcium, this is plus calcium, troponin is not shown, but it lifts up the tropomyosin, and now the cross-pitch cycle can actually happen. So here you can see actin, calcium plus, actin, tropomyosin, and tropomyosin. And so this is that you have the different names with that. And when it goes without calcium, it goes into the actin TMTN. So this is myosin binding site exposed, relaxation. So this is essentially the cycle that goes on. So essentially, what happens that if you just remember this, that when you basically try to work out or doing a strenuous activity, you should warm up. So the warm-up function is essentially allowing the muscle cells to release the calcium so they are in the cytosol for the troponin to bind them. Initially, it takes a little bit of time, but once the cytosol of the muscle cell is full of the calcium, at that time this cross-pitch cycle can go on very rapidly, because the calcium doesn't need to go back to the cerebrospinal reticulum again to release, go back and again release. Once they are all released, it's in there. So if you have a very rapid movement or a requirement of this, it will just go on and off, essentially. So that is the mechanism of the myosin, of the troponin and the tropomyosin. And this is essentially helps that when the muscle is relaxed, accidentally the myosin head cannot bind and start contracting the muscle. That's basically the regulation. So this actually tells the sliding filament model, which we already talked about. So this is a little bit three-dimensional compared to that. So you have a Z-disc. Z-disc, this is an entire sarcomere, and this is your plus, minus, N. And right here, this is essentially the length of a relaxed muscle. So your A band is the length of your, right here, this is the length of your the thick filament of the myosin. Right here is the I band, which is not overlapped, only acting, not overlapped with the myosin. Now you have the ATP and the calcium, and now it contracts, and see that what is happening. This I band is short, almost disappearing. This I band was thick, is almost disappearing. Right here, this middle part is also the H zone. It's not mentioned in here. The H zone is also disappearing, essentially, but the length of the A band remains the same in all cases, because that is the length of the myosin. It never changes. So the length of actin doesn't change, the length of a myosin doesn't change. It's just the degree of overlap between the thick and the thin filament, which is a thin filament is the myosin, thin filament is the actin, thick filament is the myosin, and the degree of overlapping just changes on this process as well. So essentially, let's take a look at one sec, because I think that it's probably a better idea to take a look at one of these. So let's. Sure. Don't worry about this part. This is a signal. So the next part is that how the cross bridge. So these okay. So essentially that's the process. And although I showed this, these are in your supplementary videos. So take out supplementary videos because none of these pictures actually do the justice. Essentially, if you just look at the animation one time, it just sticks to you much better. So definitely take a look at those. So one of the last part that what we have is a role of myosin 2 in non-muscle cell. These are all myosin 2 that is contracting on those. So contractile ring during the formation of two daughter cells from one cell that is in there. So sometimes what you can do, that you can take anti-myosin antibodies and then you can inject a living cell with those antibodies. You cannot just diffuse them. Antibodies cannot pass through the plasma membrane, you know that. So these antibodies can go into the cytosol and they can bind to these myosins, essentially. And what you're going to see that you end up with a multinucleated cell, because after the cell division, you have multiple nuclei. So nuclear division is perfect. There's no problem. But the cell cannot divide due to the formation of a nuclear ring because there is no myosin associated with that. We are going to see that the epithelial cells, they are next to each other in your lining of your blood vessels, lining of your intestine, everywhere, body lining. And we have seen that this is the apical parts of the cells, basolateral cell, and each one of them is attached to the next one with the adhesion belt. And this adhesion belt, the tight junction is close to that area as well. It's just a specialized area of the adhesion belt. If you don't have the adhesion belt, say for example, when food is moving through, or even the blood is moving through capillaries or arteries, just the sheer force of that will take these cells out. So these adhesion belts actually keep them still, you slough off many cells, thousands of cells every day, even with that. But still, this actually keeps the cells together essentially. And we're going to see that folding of this epithelium during embryogenesis, that how it helps in this process. And also these are the fibroblasts, when just mechanical stability that across the cells, this also increase, and these are also help in keeping the stability of many of these cells when you even just do this in the culture. So these are some of the examples of that. All right, so after that, we'll just quickly go over this intermediate filaments. This is the next part. Now, you can see that you just probably think it's like why there is so many of these things, everything, why it is needed to be highlighted. The reason is that each one of them will probably appear in your exam as a distractor or a part of the question, each one of them. So that's why just make sure that you put a triple star in there, that it can withstand mechanical stress, not dynamic, display similar cellular distribution. They are not directional, no plus minus n. Filament assembly does not require ATP or GTP. They are stable. They do not have any associated motor proteins and help in the cell's motility. So all of them are super important. So definitely remember that. Then we are looking at the organization. Just remember just only one step other than that. So you have a single protein, and then it forms a parallel dimer, and the two parallel dimer forms an anti-parallel tetramer. So just remember that for parallel dimer, there is an anti-parallel tetramer. This is the only step that is a little tricky, that just one side you have the head, one side you have the tail, and the opposite. Then they form the protofilament, protofibril, and then forms the intermediate filament as well. That's it. All right, so here what we are looking at, the major classes of this. So you have all these different proteins, acidic, basic, keratins, desmin, GFP, those. These are all self-explanatory. You do not have to memorize those. Now, this acidic and basic keratins, remember that why we name them. In the acidic keratins, you have the amino acids which are more acidic, glutamic acid, aspartic acid, they are more in there. Whereas in the basic keratins, there are asparagine, there are the basic amino acids are a little bit more in those keratins. That's why they are called acid or basic. They have nothing to do with any pH of the cell or anything with that. And there are neurofilaments. They are neurofilaments like a long, medium, and high, essentially, many of them. These are also lamins, which is inside the nuclear membrane. So these are all different proteins and different distribution, and these are some of the proposed function for these as well. So know these very well, and these are some of the glial cells in the neuron they are supporting, and these are your keratins in your epithelial cells as well. This gives you huge amount of this abrasion resistance, essentially. All right, so finish the part 3 and we should be good. So start practicing